Manisha, and today I'll be giving a short presentation on doing pseudobulk analysis with using the pseudobulkDGE function from the scRNA package. So for today's presentation, like the main objectives that I will go over is about what is pseudobulk analysis and why it's used, learn about the basic steps of pseudobulkDGE using the OSCA guidelines, and also compare the existing registration pseudobulk function from the special LIBD package that we all are using versus the aggregate cell across cells plus the pseudobulkDGE workflows. And then just understand about the input parameters, the steps involved in the pseudobulkDGE analysis, that is normalization, filtering, and output. So to start with, what is pseudobulk analysis? We aggregate the single cell or the special counts into different group level profiles where we treat the bulk samples as groups. And then this pseudobulking basically is suitable for doing the differential expression analysis. And here in this figure, which is from the transcriptome special gene expression paper from the human DLPFC, which is the human pilot data analysis. So in this particular figure, it shows the process of pseudobulking. On the left inset, we have the histological image where we have all the different cortical layers on the BZM slide. And then when we do the pseudobulking, we are basically taking the counts metrics. Here we have in the middle figure, it is representing all the different colors are representing the different layers, layer 1 through 6, the gray matter layers and the white matter. And then for each of these, the sum of UMIs are calculated row-wise. And then we do it in the pseudobulk counts metrics, where we have the sum of UMIs, the layers, and the genes. So then, having an idea about pseudobulk, what is pseudobulking, let us look at the OSCA workflow for pseudobulk analysis. So the first step that involved in this is aggregating the counts, that is making the pseudobulk metrics, normalizing the counts, filtering the lowly expressed genes by using the filter by expression function, and then fit the models using different methods such as jar or boom, and then extract the differentially expressed results. And then following this, we can interpret the differentially expressed results, interpreting the log fold changes, the p-values, the FDR values. pseudobulkDGE, we have this OSCA workflow for pseudobulk analysis, but scRNA gives us another function called a pseudobulkDGE, which actually automates the whole pseudobulk differential expression pipeline. So it is basically a wrapper function which involves all the steps from pseudobulking to normalization, filtering the lowly expressed genes and everything. So this supports the AJAR method, supports two methods, AJAR and boom. And for the input, we give a summarized experiment, the different labels, design, coefficient, and condition. And as I mentioned, it handles the normalization, filtering, log counts, and log counts. So just as an overview of what the pseudobulk workflow looks like, we give the input as the summarized experiment object with the counts. We define the groups. That is, for example, in our special analysis that we do, we can define the labels, we can define the groups based on the base space domains. Then aggregation, some counts within the groups, that is, we are pseudobulking. pseudobulkDGE normalizes the counts using the calc norm factors. And for filtering, that is removing the lowly expressed genes, it uses the filter by expression. Then it also does modeling, it fits the linear model, estimates the dispersion, and then it computes the statistics, differential expression statistics. And as the output, it gives a table of differentially expressed genes, log fold change, and the p-values. For the normalization and filtering, as I mentioned, it fetches the calc norm factors function, uses the calc norm factors function, and then filtering, and then the log counts after normalization, it generates the log counts per million log counts metrics. Now, why we are looking at this method, this wrapper function of pseudo? So, and why not just use, and why, why we can use, why compare it to the existing registration pseudobulk function that already exists in the special LIBD package? So, one difference is that registration pseudobulk applies the filter by expression criteria, the filter by expression function across all the spots, which the which might remove the genes with overall low expression, which might be expressed in different special domains that we are looking at, different base space domains that we are looking at. We might have situations where we have limited sample sizes, or we are more interested in looking at domain-wise expression. So in such situations, instead of using the modeling results from registration pseudobulk, in such situations, we can pseudobulk the samples separately using the aggregate across cells function, and then use this pseudobulk data in the pseudobulkDGE function. So this goes as an input to our pseudobulk function. So just again a brief a recap of this pseudobulkDGE function, that it performs the differential expression analysis on pseudobulk data. Main arguments to keep in mind is the data, the label, that is the grouping variable, which can be the different clusters, for example, the base pair clusters that we look at, and then design. So here in the design, you can give the formula from looking, which is like equivalent to the step-by-step analysis in OSCA that we follow, and give the inputs as give the input to the model metric. So instead of writing as model metrics, we just give the design formula, what is our covariate of interest, and followed by the different covariates that we are looking at, could be age, sex, PMI, and other different covariates that are important for the study. So looking at this, so when you look at the model metrics, it generates the coefficients that it generates, and that, like, you can take a look at those coefficients that are generated, and then based on that you can decide your coefficient of interest. And for example, if you have a study where you're doing cases versus control, in that case the coefficient of interest could be looking at the case. It could be any disease like Alzheimer's, schizophrenia, autism. So in that case, how those coefficients are defined, that you can take a look at from the model metrics. So this model metrics, it will generate a table which has like the intercept and the different columns that are generated from the inputs that we are giving in this particular formula, like diagnosis and the different covariates. So this just as a information to remember, that the coefficient that we are looking at, coefficient of interest, the name we can get from the model metrics. And then depending upon the condition, what is the primary condition that you are looking at, could be, as I gave an example of cases versus control. So in that case, the primary condition that you can look at is the diagnosis condition. And then the raw data is the gene level metadata. And then you can you can tell the function which method you want to use. It could be HR or it could be. So based on this, I just have small demo script to show. Okay, so so first I will just like show an example of which is given in the scRNA package. I will just try running that particular package as an example. So in this, basically the first is including the library single cell experiment and summarized experiment. Then as an example, let us set the seed, and then we call the library scuttle, and then let us make a SE object, like just making a mock SE object, and then pretending that we can pretend as if we have like 8 samples. And then to this particular SE, we can make up like few clusters and also add some counts to this particular SE object. And then after having added these clusters, some information we can add based on like what we can define a data frame which has the samples, a sample column from the SE, and then the clusters that we just created. And then as a first step, we do is like aggregate across cells. So for aggregate across cells, the input is SE object that we just generated, and then the info from the data frame. So we pseudo bulking at the sample level, sample versus domain level. So this is what we have. So we have created the pseudo bulk object using the aggregate across cells. And so, like, just for the purpose of demonstration, let us make an experimental design with the 8 samples. So here, say suppose we want to look at some variable or some experimental design for any kind of drug. So just creating this variable, and then and then for the differential gene expression analysis for the pseudobulkDGE, the input that goes in is the pseudobulk object. The label is basically the clusters that I mentioned previously, and then condition here is the drug, and then design here is your variables that you want to include. So like say for any experiment which is focusing on some kind of drug, so that could be the column name that you can include, and plus the different covariates that you're interested in could be an addition to this. And then the coefficient, say drug 2, which is just an demo name, and then we can just kind of so then this basically generates like it will generate a list of 3 because we created like 3 clusters. So in pseudobulk, it basically goes across each of the clusters to do the analysis. So it goes cluster by cluster. That is why we have in this, we see this as a list of 3. Right now, it does not have any information but linked to it. But for if you have the special experiment object properly loaded, then you can have, then if you look at each of these names, then you have like different columns of log fold change, p-value, and so on. So in our group, just for example, how I am using this particular function is, I will just show a demo for that as well, so that people can relate to it. So I am creating, I'm just creating like small demo SP object where I am defining the number of genes, samples, and number of domains. For example, I have like 100 genes, 8 samples, and 9 domains. And also then generating random number of numbers per cell group. So just for the reference, so total number of cells, and then let us see how many total cells we have. In total, we have 822 cells. And then I'm creating this counts metric just for the demo purpose, where I'm giving the row names as the genes and column names as the cells. And then I'm just expanding this sample and domain IDs as per their group. So basically in the sample, for each sample domain, I will have sample IDs S1 to S9 because S8 because I just made 8 samples over here and 9 domains. So let us take a look at the sample IDs, just make the sample IDs and domain names. And I'm just like creating a mock data for just a metadata which is a data frame including the sample ID. So I'm like the way we get the base space results. So like after doing the harmony correction and everything, I just say we have this column called as base space harmony K9, which includes the information of base space domain at 9, and then the diagnosis column, which has the control and the case study, total cells, and then just age and PMI. So with having all this information, we can just like create mock single cell experiment. So this looks like, so this is like a single cell experiment, 100, we named 100, sorry, we had like 100 genes and 822 total cells. So that is why dimension is 100, 822. And then we have the counts information, row names as the gene names, column names. So, and then call data column names as. So just we created the sample IDs, base space, harmony, K9 column, diagnosis, age, and PMI. So just mock single cell experiment that we created. And now for the main analysis, differential gene expression analysis, pseudobulk the data. So here, this particular way of writing, so for the aggregate across cells, I'm giving the SP as an object, and then in another parameter that I'm passing is the like in the form of data frame, because I am looking at base space and I want to aggregate the data by base space and the sample IDs. So this will be, so when this data is generated, it's basically sample special domain times the number of samples that you have. So this is like we had 8 samples and 9 domains. That is why if you see the change in the number of dimensions, this is 8 into 8 times 9 is 72. So we have the dimension 72. And the difference, if you notice here, is colony colony. So here we have 3 additional names that are added. So because we named this as base space and sample ID, I did not want to touch this thing. We can just rename this as registration, just for the purpose so that we don't have a confusion and we don't touch the already existing. So you can see here, basically base space, registration, sample ID, and N cells. These are the new columns that are created. Let us take a look at how the N cells look like. So when you have your special data, so how this pseudobulking basically aggregates all the counts, and then for each domain wise, for each sample, these counts created. So we have 8 samples and 9 domains. So these are like 8 times 9, 72. These are these N cells are created, 72 N cells are created over here. And then like if you're analyzing, if you're analyzing a special data, so in that case you can, in that case you can like have a number of N spots. I should have named this as N spots. So you can call it as N spots. So I'm just renaming this in terms of the N spots. For the special data, we have the number of spots. We can filter these. We can filter, say, you have like number of cells, but minimum recommended one for the actual data is like minimum should be 10 N spots that you should be filtering. So I just want to keep the data which has more than minimum N spots. And then for prepare, and then like here is another important step after doing the aggregate across cells is you convert your variables, and basically this is preparing your data for the pseudobulkDGE analysis. So here I am just, in case of my different variables that I have, the different covariates that I just made in this special experiment object, is I am just converting them as a factor. So for the diagnosis, I have case versus control. So you can just convert that into case versus control. Age and PMI could be numbers, so converting them into numeric, and then base space as a factor. So we have the levels as the different base space domains. So we just like convert the data and prepare it for the pseudobulkDGE. And then for running this particular function, run pseudo. So here the input goes as the data, that is your pseudobulk data. design coefficient and what method you want to do. So this is just run pseudobulkDGE function. I just wrote a function in the form of function. But what this basically has is a pseudobulkDGE function. So here you can create different models for your data. So by models, what I mean is you give the data the label as the base space domain of which is the domain of interest in my example that I'm showing currently. Here in the design, as I said, these are basically the different covariates that you're looking at. So imagine this as how you want to design, how your input would go to the model.metrics function. So these are the different covariates that you're looking at. In this case, I just have diagnosis, age, N-spots, and PMI. So say my very first covariate of importance is diagnosis. So I'm looking at diagnosis. So that will be my very first variable that I'm giving in the design formula, followed by the covariates. So because I'm keeping this, the covariate of importance should be first. So imagine how the model matrix creates the different columns. The first one is the intercept, and then the second one is basically the first element, the first covariate that we are giving to the formula. And then you can experiment with this, writing this formula. It can be an interaction model, or it could be just testing different variates altogether. So this is an important step. So that is why I had written this. I wrote it in the form of a function that I can just call here. And then in this case, coefficient, as I mentioned. So if you look at the model metrics, how your variables, how your columns are named. So in the model metrics, you can, if you take a look at, I mean, like I'm giving diagnosis as the first variable. So the way it would be created is looking at cases versus control. So the column that would be created, for example, could be like diagnosis case. Here by case, I mean like different diseases or different experimental designs that you're testing for. So that could be one important thing that you can do this. So, and then you just give it method that you want to look at. So by method, it could be either HR or WOM based on the experimental design that you're looking for. So in order to reach to like the step where you're deciding what covariates or how to write your design formula, I would say that after the pseudobulking the data, it would be important to take a look at the different covariates, exploring the different covariates. We also had like a previous session where we talked a lot about exploring the different covariates, exploring the data. That is very important step, and that's how then you can decide what covariates go into your design formula. So this is just an example. So here this would just like this particular thing would run into an error because here the SP is not linked, but like if you have your SP properly created, then all your information will be just like your analysis. The pseudobulk variable, the pseudobulk data will also extract the information from the SP, but I think I did not do that in this case over here. So yeah, this is how you would use the pseudobulkDGE function. This is what I had for everyone today. Yes, yeah, that's it that I have. Thank you.